So warm welcome to everyone, and we are really happy to have our chief guest today with us, respected Padma Shri Professor K. N. Ganesh sir. And without taking much of time, we will be starting this event. And I request all the dignitary to please be on dais. So I request Professor K. N. Ganesh sir, Professor Bhushan Patwardhan sir, Dr. Manohar Furtado sir, uh, Head of the Department Mahesh Kulkarni sir, yeah, and Dr. Ramanna sir. So we have gathered today uh, to remember the great contribution of respected Professor Vijay Khanna sir. He was one of the pioneer for drug chemistry subject, which is first time in India, like where you can combine organic with biology and at the same time with pharma. And he has contributed in an immense way and made generations of chemists who are really expert in making molecules which are really saving a lot of lives. So without taking much of time, I request Dr. Ramana sir to please felicitate all our guests and welcome them with floral bouquet. So first I request please Professor Ken Ganesh sir, our chief guest. Professor Bhushan Patwardhan sir. I request Mahesh Kulkarni sir also. Dr. Manohar Furtado sir. And also I request for Dr. Mahesh Kulkarni sir on behalf of like NCL, because this program we are coordinating like with collaboration with NCL Pune, at the same time SPPU along with Ahmednagar College Alumni Association. So with this, I'd like to mention that considering the limited time, I request Dr. Ashok Giri sir on behalf of NCL, please express few words about the program. At the same time, I would like to mention Dr. John Barnabas sir, having long association with NCL and with Ahmednagar College, and sir will explain about it.
So, good evening all, respected dais, my dear friends. On the behalf of our director, I would like to welcome all of you, and particularly Dr. Ken Ganesh, Dr. Bhushan Patwardhan sir, as well as the program that he wants to do is Dr. Manohar Furtado sir. So it was unfortunately there was another program in parallel. We booked actually our main auditorium, but for some reason we have to shift. But still, I feel nice to see all our friends, old friends here. So I don't want to take everybody's name, but welcome to NCL, all of you, on behalf of NCL. Actually, this is not for the, especially I see there are a lot of students coming from Ahmednagar College. So I just want to introduce a brief, although you have a very nice trip today around the NCL, seeing our archives and other facilities. So NCL is a CSIR, Council of Scientific and Industrial Research Laboratory. It's a premier research lab. So we have at any given time around 400 to 500 PhD scholars, as well as equal number of project associates and other project staff. So we are with a very subsidized rate, and we also provide the accommodations. So this is particularly I'm saying because there are a lot of MSc students and BSc students here. So, so that's about National Chemical Laboratory. Actually, I was unfortunate that when I joined NCL that time, Dr. Barnabas, John Barnabas sir was not anymore there. He passed away in 1994. I joined NCL as a research associate in 1995. But since all my colleagues say that since he was head of Biochemical Sciences Division, I should talk a little bit about Professor John Barnabas. So actually, the most of the source for his information is coming from one of the student who was my guru, Dr. P.K. Ranjekar, Prabhakar Ranjekar. So he was his fourth PhD scholar seems, and he was very popularly known as a John Sir. I think all the people will remember. And most of his career was in Ahmednagar College as he started this biochemistry course, and he was from lecturer to professor there. But then he moved to, and I mean historically, I think all of you know that he has published very high impact papers, including Nature in 1971. And I think we have seen this instrument where he did some experiment at Nagar College in the PG Center of Biochemistry. So he used to work on this evolutionary genetics, and particularly a sequence of albumins and other proteins, which are actually quite conserved in higher organisms. And when he moved to NCL as a Jawaharlal Nehru Fellow, and then became head of Biochemical Sciences Division, and even when he was even in Ahmednagar College, one of the things which prominently told by Dr. Ranjekar to me is that he used to bring the stalwarts not only from the nation but even outside the nation. Like Nobel laureate came to Ahmednagar several times. That's what I came to know. Even Professor Talwar, I mean, senior people will remember Mukherjee, even Professor Srinivasan, Note, Professor Verma. All these people he used to come to the Ahmednagar College. And more interestingly, he used to have this very famous conference, which was more of a formal and informal. A lot of students used to get involved into it. This was typical like at international level, something called Gordon Conference, or maybe in India, there's something Goa Conference. So it was similar like that Mahabaleshwar Conference, and it used to be like very informal, and I think several of senior people have benefited to interact that time with, I mean, one of the best or fine biochemist and chemist and biologist that time. And this used to be at Mahabaleshwar, so that means the student have access to these stalwarts to interact formally as informally. And this is something I will say very special. It is very important to interact or get time to discuss with the stalwarts. Like today we have Professor Padma Shri Ganesh sir, Bhushan Patwardhan sir, and several of you. So these opportunities actually students should use to interact, talk to them. So our idea we are discussing with Dr. Manohar that can we start this Mahabaleshwar seminar. This is a very crude idea. I thought we should do it because this will be largely benefiting to the students. And I hope we plan to do something next year. And we, of course, expect support from all of you to participate as a mentor or as a students also should join. So we are seriously thinking about it. I can't really say anything. Probably Professor Manohar sir will say something about it, what is his plan in mind. So with this, I welcome you again. Thank you, sir, for your time. Thank you, Patwardhan Sarji, for coming over this occasion. My colleagues, Dr. Ramanna and Mahesh Kulkarni, gave me this opportunity. Thank you. Thank you.
Thank you, sir, for insightful information about NCL as well as the way we had a long tie-up with Ahmednagar College through Professor John Barnabas, sir. So if you remember, actually, this event has started since long time back. When we had a first discussion during the lockdown, as an alumni, we should start something. And before we start, like as a young alumni, our seniors already started thinking about that. And that's how Professor Bhushan Patwardhan, sir, Dr. Manohar Furtado, sir, they come up with and they have started Science Education and Research India Association. And I would like to mention that this is second orientation like oration for this particular program. But the first was conducted by Professor Bhushan Patwardhan sir in Ahmednagar College, and now this will be like on a rotation basis at different institutes. So to take more information, more insightful, I'd like to request Dr. Manohar Furtado sir to please explain about this new NGO and what will be the next plan.
Thank you, Dinesh, for the kind introduction. I'd like to thank all of you here. I'm not going to name everyone for taking time out to come here and be with us and give us access to your intelligence and discussion. So, and I also want to thank Ashok Giri for saying a few words about John Barnabas, so I don't have to discuss him here. And lastly, I also want to thank him for going over a few of the programs in NCL, which I think would be useful for students. We want you to continuously access their websites, find programs, and apply for internships, scholarships, or other TA positions or whatever is available in here. So a couple of years ago, I was working full-time till about 2024 June, and then I retired. And then I'm thinking, what can I do? So I looked around and talked to some friends of ours like Dr. Bhushan, Dr. Lonkar here, had some discussions with Dr. Khanna as well, and came to the conclusion that we would like to do something to help the students and also to some extent the faculty, and provide instruments and reagents to the extent possible to the John Barnabas PG School in Ahmednagar. Many of us graduated out of that institute, and we're all grateful for that. So last year in August, we officially registered a trust with the Indian government called Science Education Research India Association. It's a Section 8 company with tax credits, 12A clearance, and 80G clearance from the Indian government. So it's officially there. We can collect funds, and we have been fortunate enough to get about 30 to 40 lakhs into the account. We also got a 15 lakh grant from the Chromatographic Society of India to buy some instruments. So all of this over a period of time, what we want to do is build an endowment fund, and that can be used to help or fund student scholarships, student merit awards, instrumentation. We already put about 12 lakhs of instruments into the college in there. So we hope to run this for the next 10, 20 years. Not me, but somebody else will take over. But we're trying to set up an endowment that will last for a very long time, not just 1 year or 2 years. So that's the long-term goal of this. Hopefully, with the current regulations, in 3 to 4 years, we can get foreign currency regulation clearance as well. And if that happens, there are a lot of people who have graduated out of Ahmednagar College, not only in India, but working in big positions, especially those who passed out in the 80s and 90s are at very high positions, even in the US, and they're all willing to help. But we also look to you to help in any way you can, and that includes taking some lectures. So we conducted last year a 16-lecture course on quality and regulatory affairs required in industry. The goal ultimately is to prepare our students to be ready to go into an academic institution or an industry, depending on their choice, but have the fundamental training required to make them ready for it. And not all of it is in your syllabus. So like last month, we got Dr. Girish Tillu from the health sciences to conduct a course on research methodology, clinical trials, and some of these things are not actually in the MSc syllabuses. I've gone through most of them. So wherever we can, we're going to supplement by inviting speakers, by holding mini symposia, or like Dr. Girish mentioned, a more bigger attempt in terms of mimicking what was done at Mahabaleshwar. We don't know to what extent it will succeed, but we're going to try. So those are all the goals we have in mind. And again, like Dinesh said, we'll look to all of you for your support, continuing over the next several years. Thank you all for coming. I know many of you are past students. Some of you are faculty here in NCL and maybe not directly been in Ahmednagar College, but I'd like to thank all of you for coming. And with that, I'll hand it over back to Dinesh.
Thank you, sir. just because there are many students are also attending, I would like to mention that at NCL also we are having skill development programs, internship opportunities. And Dr. Rajesh Gondari sir is the in charge for that program, and we will be coordinating some of the activities along with Nagar College to help the student to get into the NCL. So with this, I would like to request our another guest, Dr. Bhushan Patwardhan sir. He is man from our home because he has spent a lot of time with Khanna sir. So he's the right person to say like all the details what he has about Khanna sir and his journey. So generally this name doesn't need an introduction. Like he was a UGC chairman, he's a Ministry of Ayush person, at the same time started Center of Health Science, a lot of contribution in drug chemistry also.
Thank you, Dinesh. And thank you, NCL, for organizing Dr. Vijay Khanna Memorial Oration here. And we have probably the best person to deliver the second oration in his memory. Dr. Devbakar, Mr. and Mrs. are here. Please come ahead, sir. Everybody, as we know, we try to find purpose of our life, And during student days, we have different purpose. When we retire, we still try to find out. Not that everybody finds it to satisfaction, but here is a person who found it, And he is Dr. Manohar Fatado. After retiring and after excelling in US, after teaching in one of the top universities, and also contributing to establishment of some of the top biotech companies, he decided to give his entire time, resources to bring back the past glory of Ahmednagar College, now known as Professor John Barnabas Postgraduate School. Many of us are students passed out from that school. Dr. Barnabas, as Dinesh said, John sir, to all of us, I would like to pay first my respect on behalf of all of us to him. Dr. Vijay Khanna actually is the first person whom I met when I went to Ahmednagar. During that time, students used to give away admission, even we get it in Pune University departments, and opt for Ahmednagar College, especially for biochemistry. I was one like them, And when you go to new town, you make new friends. So the first person I met was Vijay. Such a lovely person. And since that time, we got connected like brothers. We worked together on projects also. And since that time, we had decided that we will do something, related to drugs. I'm happy to see Vijay's family also here. And Vijay's home was also like our home, We used to go anytime to Tarakpur and have nice Punjabi food and get a lot of love from all his family. Vijay is a very unique person, so caring for everyone. And he used to be a bond between organic chemistry and biochemistry. All the two departments, but bound by love of Vijay. For my PhD, I also worked at Hafkin Institute, where he was a frequent visitor. Dr. Manohar also had visited me at Hafkin. And since that time, as I told, we were talking about drugs and drug chemistry. And after his graduation, his doctorate, and at that time, let me tell you that NCL was like our second home. For all university students, NCL, or for that matter, Ahmednagar students also, NCL was like our second home, And so, frequent visits to NCL. Most of the time we have spent in NCL, Dr. Ganesh, I still remember I doing my own project in the basement with Dr. U.R. Nayak, then S.N. Kulkarni, then Ravi, and then many of them. You also, I remember meeting you several times at NCL. Later, he had so many opportunities. After completing PhD, we met Professor Malcolm Hooper. At that time, structure-activity relationship was relatively new, and Professor Hooper was working on anti-tubercular drugs, and Vijay got connected with him. And actually, Professor Hooper, I remember, I still have that picture when he visited at Tarakpur. We all were at home of Vijay's. So he went to Sunderland. He did work with Professor Malcolm Hooper. Then he got an opportunity actually to work with Nobel laureate, and that time was also, he was in continuous touch with each other. Let me tell you that very few people will take this decision, and Vijay took that decision. For family and friends, he returned from US back to Ahmednagar and started working in a small college, living all that glory and all that comfort which he had in UK as well as in US. So you can imagine the kind, and doing all that, the academic rigor was never compromised. He could publish in very good journals, and during that time we started talking about developing a program, master's program in drug chemistry. And we had several meetings, and drug chemistry was not there. Medicinal chemistry, Dr. Ganesh will remember, used to be a lot of discussions that medicinal chemistry is getting weak now in India. We must do something to strengthen it. It was at that time Vijay thought of this MSc in drug chemistry, and both of us worked with many of others, to develop its first curriculum. I still remember we had series of meetings in Pune and Ahmednagar to create that curriculum. And today I can see many young students here who have either taken that and passed out and have well placed. Many industry people, Dr. Ganesh, have told me that without Vijay Khanna, they would not have got such well-trained students to work in their laboratories. And these well-trained students came from such a modest background, from small villages, All this is a great work which Vijay did, He built confidence in the minds of people and students who are coming from underserved areas all over Maharashtra, mostly in Pune, but also from different parts, He really fought very hard when he was diagnosed with cancer, and he was a person who would never give up, and all of us, Dr. Lonkar, all of us were in touch with him till his last days, and we still feel that he is with us, He's with us with all his work and with all his memories, and we thought that we should have oration in honor of Dr. Vijay Khanna, and we are so happy that Dr. K.N. Ganesh, Padma awardee and one of the outstanding scientists of India, former founder director of ISIR Pune, ISIR Tirupati, and a person who has also taken a little different route than a normal organic chemist and went into a very strong bioorganic chemistry, and today he's going to talk on a very, very interesting subject. So I don't want to be in between you and him. Let's keep this spirit of Dr. Vijay Khanna, what he showed through his life, and continue our academic journey ahead. Namaskar, Jai Hind.
Thank you, sir. Gurur Brahma, Gurur Vishnu, Guru Devo Maheshwara, Guru Sakshat Parabrahma, Tasmai Shri Guruve Namaha. So sir has mentioned very right point. Like Khanna sir has made career of many, many students from rural area. And I'm one of the example. Like starting my journey as one of the person from Dhule Adivasi district coming to Nagar College to get a 10,000 rupees job and becoming a scientist working again with Nobel laureate and many top scientists, only possible because of Khanna sir's efforts. Like me, there are many from our alumni working in many places abroad in India and IIT and ISIRs. And with that also I would like to mention a small thing that sir has mentioned about sir as a friend. But we as a student still shiver when we even talk sir's name. So whenever we used to learn chemistry, so first thought used to come in into our minds like what sir will think if I made a mistake. And till date also when we taught our student, that quality matters, like how we teach our students the basics. When students say in interview that I don't know chemistry which they have been taught last year, we remember what we have been taught by Khanna sir in 2007, walk through the structure. we still walk through the structures. And whenever we think about chemistry, we think about mechanism, we think about how to do additions. So all these concepts, what we learn is because of Khanna sir. So with this aspect from the student point of view, I'd like to request now if Saurabh Khanna, Khanna ji is here. Okay, so we will be having like some feedback from family members also after this event. So with this, we have made one small video of a sir about his overall journey, whatever photos we have received from alumni and some of our group members. So we will show it shortly. And I'd like to request guests, please be seated. And after that, I will request Mahesh Kulkarni sir, after the video, please be ready to introduce respected Padma Shri Professor K.N. Ganesh sir. Video.
दुनिया में यूं आना, दुनिया से यूं जाना यादें 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 बातें भूल जाती है यादें याद आती है ये यादें इसी दिल जानम के चले जाने के बाद आती है
Yeah. With this, some emotional moments, we will proceed little bit forward. Uh, Everybody now, like, just trying to imagine, like, the way they have spent their time with Khanna sir. So I'd like to request sir to introduce respected Ken Ganesh sir.
Good afternoon, all. It's my privilege to introduce Professor Ken Ganesh, an internationally renowned bioorganic chemist, distinguished academic leader, and a recipient of Padma Shri Award 2023. Professor Ganesh is widely recognized for his pioneering contributions to the chemistry and biology of nucleic acids. His research has significantly advanced our understanding of peptide nucleic acid, DNA molecular recognition, DNA technology, structural biology, with important applications in therapeutics and diagnostics. His work has successfully bridged the disciplines of organic chemistry and biology, making a lasting impact on biomolecular and medicinal chemistry. Beyond his scientific achievements, Professor Ganesh has played a transformative role in shaping Indian science, education, and
keep my bag somewhere, I think. Can't just put this. Okay, thank you Mahesh, for your kind words of introduction. And I must also thank Dr. Manohar, Dr. Bhushan Patwardhan, and others who are involved in making a decision to call me for this Oration lecture. I think it's a very moving film, what you showed. And first of all, I must start with John Barnabas before I get on to Vijay. I was at CCMB Hyderabad, and John Barnabas at the time, he was biochemistry head, and he used to come frequently as a research council chairman. And he was such a fantastic teacher. When he comes, you start talking about chemistry, he forgets time, everything. Although he was a evolutionary biologist and biochemist, but he had great interest in chemistry. Many times he will forget time and used to discuss about biosynthesis, etc. And one day he told me, Ganesh, you must come to Pune. And because I used to tell him how difficult it is to get organic chemists to work for me for PhD in CCMB. I was started working on nucleic acid, that area. And he told me, I think you must come and join NCL. You will get a lot of PhD students in chemistry. And what he told me was that it is very easy to do biology in a chemistry lab than trying to do chemistry in a biology lab. I don't know what biologists here they feel. And and he and Dr. Mashelka were responsible for me to come to Pune. Although Dr. Bhargava, who was the director at that time, wasn't very happy. But anyway, I gave it. What immediately struck to me when I came to Pune was this great coordination, cordial relationship between Pune University and NCL, whether it is in chemistry, biochemistry, or whatever. People would freely walk in, and like Professor Wadia, Avinash is here, Professor Davle, many of them, many times difficult to distinguish who is from which institute. And they were great teachers. That is another thing, meeting Professor Wadia, Sohan Modak, Dilip Debakar, whether it is in chemistry or biochemistry, great, fantastic teacher. I think that really attracted me. I said that I have made the right decision to come over to this place. So mainly I came because of John Barnabas, and very frequent, although I was in organic chemistry, very frequently because of him I used to go and meet colleagues in biochemistry, Ranjekar, Vidya Gupta, and many of the other people in biochemistry. And so the idea was at that time NCL, everybody was busy in organic chemistry, working on pesticides, insecticides, etc. And terms like DNA, oligonucleotides, peptides were all Latin and Greek for the chemistry crowd. So one day, one of the professor, Dr. R.B. Mitra, who was the head, he said, we have something in common. I work on insecticide and pesticide. It has got phosphorus atom. DNA also has phosphorus. We must collaborate, he said. So that's a very, very imaginative way of, interpreting, chemistry and the connection to biochemistry. Anyway, but one good thing was that although I was not such a hardcore organic chemist, NCL accepted me. Not only accepted me, and when I even became head of the organic chemistry, did not force me to work on processes, etc. They gave a complete freedom. In fact, that's where I started in nucleic acid chemistry in India, for synthesis. And when I was in CCMB, anything I want, I will look into Sigma Aldrich catalog. And when I joined NCL, I will look into Vogel's book, how to make it. That made a great difference. And I was assigned, Dr. Nathu and Dr. Vaijayanti Kumar as my, assistant. That's what was given at that time. And it's a great time we had. And this close collaboration with Pune University, I used to go and teach. Wadia, when Wadia comes for one of these assessments, more than the student, I am nervous. The kind of questions, they would ask in such a friendly manner, not in a very thing. It's a great learning process for us. And then I met Dr. Vijay Khanna. And again, John Barnabas took me to one of the Mahabaleshwar Symposium. It was a great. I'm glad that you're trying to revive that. And it was not very easy. When you go to Mahabaleshwar Symposium, each one of us are assigned 10 students. And after lunch, you have to go with these 10 students, walk around, sit below a tree, and answer all their questions. That is a very tough thing. I'm sure the Deep Debakar and all of you must have experienced that. It's a fantastic thing, to create that. And of course, all of you know how John, published, such great papers from Nagar College, for which he became very famous. The college became, famous, etc. And later on, I had very good interaction. We started what is called the Indian Bioorganic Chemistry, for which he was the president, and we had great times. And Khanna used to come, any symposium, conference, any meeting we organize in NCL, He would be a part of the organizing committee, and I also gone and given some talks in Nagar College. I hope those who have heard me, those repeating some of them today. So he was a great friend, colleague, and what was described, the great teacher. That was important. I think Pune University and NCL, of course, had great researchers. I think it is a fantastic combination Pune had during 80s, 90s, and 2000. Fantastic people. And then, of course, later on, we had Dr. Mashelkar and Paul Ratnaswamy, Dr. Shivram. They came, and Pune had Professor Bide, and it's a fantastic thing. And I was very lucky to be living in that era to see these great personalities in both institutions. And I visited Nagar College a few times. Again, what he introduced, drug chemistry. Today I thought I will put my title as Making Drugs from Nucleic Acids. Then I thought if that happens, the Narcotic Bureau will immediately come and catch me. For that I converted into medicines rather than drugs in a light-hearted way. So both, I think, John Barnabas and Vijay Khanna, we had great interactions, and I've seen how good teacher he is. And I still remember when they designed this curriculum for the chemistry course, MedChem, then he really sent it to me, and we discussed, a few things, how it could be changed, how it could be modernized. But that was the very first thing, to introduce a MedChem course in Nagar College. I'm not sure whether Pune University Chemistry Department had that course at that time. That was a great, I think. It is what, people like this, they build institutions. And I think very good to see that many of the students from, Khanna School and Nagar College have gone to greater heights. And today morning, I received a WhatsApp message from Deepak Salunke, who did PhD here. I didn't know that he was from Nagar College. I've been interacting with him. Now he's a faculty in NIPER. He said, sorry, sir, I won't be able to come. He keeps on consulting me, and I'm unable to come. But he said, today, whatever I have achieved, it's because of what I learned in Nagar College with, Dr. Khanna. And I think your pamphlet also gives a few other great students. I think that's the greatest, thing for all of us, teachers is when students do much better than us, we are very, very happy. We are not envious, we are very happy. So with this brief thing, let me go on. And also very nice, you said that you have started this science education and research, India, this thing. So that SER, since I was in IISER, it has become reverse, Science Education Research Association of India, I think. It's a very good move, and I think must really those who are close to this concept. I think when ISIS were established, they said that the E is important. Science education, E comes before R. Just like in CSIR, Council of Scientific and Industry, that thing comes before research. It's very important. I think those who design these names, they have that, whatever their focus, policy, very well defined in the name itself. I think this is very good, and I think whatever we can all contribute, I think we should take it. It's good that you have, registered, and you have a lot following, and I hope it becomes, I think, a lot of things, and maybe Mahabaleshwar kind of symposium could be, a part of that, etc. So very pleased to see that. So today, I'm going to talk about some making medicines out of nucleic acid. So I thought that since, Khanna was so close to the medicinal, I think most of you know about that. And when all of you go and you have something, headache, you just pop up a pill, or something else, you pop up a pill, and it gives you great relief. And for a moment, if you think back, what has enabled you to get this one drug, put it in, what effort has gone before that, before making the drug available to you, the medicine available to you, it's a great story. And I'm sure if the MedChem course, I'm sure it's maybe still going on, and how a new compound is, discovered. So there are various stages, any drug from the discovery stage, I will make it very simple, I will not take too much time. So from the stage one, when you discover, in the lab that it has got biological activity, there are several assays, etc., to the finally, if it gets approved as a drug, it may take about 15 years. I'm sure all of you know about it. And it is not very simple. People who are involved here will never be involved there. So it has to go through rigorous evaluation procedure. So the first step, how the new molecule is that, people start with lots of compounds, This is a conventional way, but now today, in fact, people are talking about AI, ML, how it can simplify that. But otherwise, you start with a pool of about, say, 10,000 compounds. finally there is a chance that one compound may receive regulatory approval, one or two things. So rest of them, they may not, they'll be thrown out of different stages. And how do we get this? Either you can start with a lot of compounds, or you can start with, which is natural product, synthesized compounds. In fact, at NCL, we had a great program of collecting all compounds synthesized and just keep it, and then keep it for screening compounds. That was the program. And early stage of drug discovery. And after that, once you know that some compounds have biological activity, let's say, from 10,000 you come to about 250 compounds which may have biological activity, then you go about optimizing the structure, thinking what is the biological activity, chemists make analogs of them and see how you can improve the activity, and finally get to some compounds which are called as lead compounds. You make it about 5 compounds as lead compounds. And then you do all this clinical development, you give it on, animal studies, then go to humans. In humans also, it is first not directly given to all the patients. It is given to healthy males, and then it is given to the patients, male, and then it goes to women, and then to children. So when something does not perform satisfactorily, it is all dropped out, and you go to the next, one which survives all these things, and then go to clinical development. Then you have to make formulations, and all that. Manufacturing techniques have to be done. So many things happen simultaneously here. Should I go there? I'm blocking, perhaps. I'll stand above. I think it's better. So you have, all these clinical studies, and then finally it is filed, and there are regulatory approvals, etc. And one other compound may come. So this process involves lots of things. I will not go to how from a hit to the lead compound optimization, and all that will happen. It's a very complex process. It takes 15 years, and also it consumes a lot of money. And it is not done by a single individual. Various institutions are involved with many institutions and organizations. Basic research is done, the idea generation in universities, institutions, government labs, etc. The moment you go to the discovery, the compound, many companies will come in, biotechs, pharma, because they know what is it like, for the administration purpose. Preclinical development and exploratory development in all these places, a lot more chemical companies will come. The academic medical centers, hospitals, clinics, etc. is done. So this 10 to 14 years will involve not only that, the cost would be about $1 billion. That is estimated, take it this way, that way. Why does it require so much? Because you are paying for all the failures, because whether you get 250 compounds, 300 compounds, when you do clinical development, the costs increase, particularly when you come to the clinical trials where humans are involved, a lot of ethical clearance, insurance will be, you have to give to them, we have to follow it up for term, etc. So it takes about $1 billion to get, one compound. Nevertheless, it's patented. So initially when drugs are introduced, the cost will be high. Over the years, it will, come down. Now, what are these drug molecules? Now, these drug molecules are all, as most of us know, they are small molecules, about 500 to 800 molecular weight. They are all small molecules. You know about a lot of antibiotics which you take, penicillin, ampicillin, erythromycin, streptomycin, or these are all, many of them are natural compounds, but then you have synthetic compounds, ranitidine, anti-ulcer, cimetidine, etc. Then you have antiviral compounds. We know, all of you, at least in your own life, you have seen, the COVID, a lot of antiviral compounds had to be developed, or HIV. And then you have heart drug. So these are all synthetic compounds. So you have both natural compounds and synthetic compounds. Then you have several anti-cancer compounds. Some of them are naturally occurring, and naturally occurring ones may not be good. You have to modify them, etc. So these are all the small compounds which have become drugs and which have increased the life expectancy of, average human being. Now, all these molecules, what you do, they must go and bind to a target. And these targets are normally proteins, or nucleic acids, etc., which are macromolecules. So these small molecules, I will not go into too much into chemistry. For example, a DNA, the double helical structure is replicating. So many of these molecules, each one, they have different mechanisms of interaction with the DNA. They can intercalate, they can bind in the groove, or they can, be non-covalent. They may produce some radical species which go and cuts. Some of them may bind to the replication site. So all these things, have been done, at least in case of DNA. But the question to ask is that, when you go to proteins, it is not that easy, because they go and bind to an active site, and if you want to sort of design a new drug which is binding to that particular active site, it is very difficult. A lot of computational thing, because we don't know how a protein folds itself. So it's not very easy. In fact, very few entirely computer-aided drugs have come out. But now that new programs have come for which, we have, people have, the Nobel Prizes have been given, AlphaFold, they predict, a lot of powerful computer programs have come which can predict how proteins fold and which are the targets, what are the commonality it is happening. I think the first drug based on such a thing was recently an antibiotic had been, approved, etc. On the other hand, it is much easier to design drugs which can bind to nucleic acids because they bind to these, kind of structures, and they're aromatic, non-aromatic, much more is known about that. So these things with DNA, nucleic acid as a target for a small molecule drug, that is very well known. But can nucleic acid themselves can be drugs? That's a question. And if so, what are the kind of things, they can do that? That's because most of the drugs for proteins are drugs or proteins which are gene products. Many diseases, the simplest definition is that the protein either is not produced in enough amounts or the protein produced or the enzyme is defective in nature. So you want to stop the activities of those proteins. So you put a drug inhibitor which goes and binds to the active site and thereby it stops the activity. That's the general principle, the simplest definition of diseases. So many diseases we know which are the proteins which are causing, which are the targets which you have to inhibit. Not all the cases, but that is known. So they all go and bind to, regulate the enzyme activity or facilitate signal transduction, etc. So many of these proteins, the gene products, it is known, it's a very well known diagram. They say that enzymes are 47% of the drugs for which, for the molecules for which drugs are designed. Or you have what is called the G protein coupled receptors, about 30% of them, and channel 7%, transporters, nuclear receptors, etc. You see the DNA target is only for about 1% of them. So all these targets are gene products, proteins. And I think all of you have studied DNA to RNA to proteins. Every protein has a RNA and then a DNA. So every protein is a gene associated with it. The simplest question to ask is that why should we allow the defective proteins to be produced first of all and then put a drug molecule to stop that activity? Once we understand this basic molecular dogma of molecular biology, can we go back and see that the protein itself is not produced? So we know that DNA makes the RNA, messenger RNA, and conventional therapy involves it goes, binds to the ribosome, messenger RNA, and produces proteins, and then you put traditional drugs and you inhibit them. So why do we go through this process? Is there any way we can stop the production of the protein, or even you can repair the protein also? That is through genetic engineering it is done. So the alternative way of looking at it is that if you can stop the production of this messenger RNA itself, then there won't be any protein production. How can you do that? How can you stop messenger RNA producing? Now, you know that the messenger RNA goes through ribosome. One mRNA, it goes into the ribosome, and one mRNA can produce as much as 200 molecules of proteins. It goes, comes back, ribosome, comes back. You can produce 200 molecules of proteins, and you require 200 molecules of drug molecule to prevent that. On the other hand, you have one molecule of messenger RNA. If you put something, you can stop the production itself, then with one molecule of drug, you can stop the 200 molecule of protein. It's a very simple, economics thing. And how can you do that? It is very easy to design a drug to bind to nucleic acid, RNA. We know that the human genome has been sequenced. So we know which gene codes for which protein is for the disease, etc. We know that. So if you can somehow stop the production, and the easiest way is, we know that one RNA or DNA recognizes a complementary strand. So if you want to inhibit this RNA, the sequence, you make a complementary strand, the one which is shown in the red, you put it, it goes and binds to this messenger RNA, and it forms a duplex. We know that complementary recognition happens, A, T, C, G recognition. So given that scene, once you have a double strand, this ribosome cannot go through double-stranded DNA. It requires a single-stranded RNA. It cannot go through RNA, DNA, or RNA-RNA hybrid, and the ribosome stalls and the protein production stops. So it's a very easy way, theoretically, if you look at it, all you require is now this antisense strand, what you are putting, that itself is a drug molecule. It's not a small molecule. A short piece of DNA itself is a drug molecule now. So no protein, no production. So whatever a traditional drug would do, this would do the same thing. A short fragment of DNA or RNA itself is a drug, which means you know different diseases, they have different genes, you know different sequences. So you simply change the sequence of the drug, it becomes drug for another disease. So very simple, straightforward, which means by this principle you can address several diseases, you know. If you go for the conventional drug, you know, some of them are terpenes, some of them are alkaloids, some of them are aromatic compounds, and different toxicity profiles, etc., etc. But here, there is a generic general structure. All you simply change the sequence, it can become drug for another molecule. So that is it. So somewhere in 2001, in 1998, I will show you the first drug based on this principle was approved, and this Times article carried in 2001, 22. And they said that drugs of the future, amazing new medicines, you know, will be based on, that's what is important. You know, all future drugs will be based on nucleic acid, the future drug, because based on this principle, the DNA itself will become a drug. So that came in 2001. Not only that, this concept has already attracted 4 Nobel Prizes. 2006 for siRNA, CRISPR-Cas, all of them are based on this principle. mRNA vaccine, all of you know about the COVID thing. Now very recently, about 2 or 3 weeks back, a vaccine for flu, which is very running all around. So mRNA-based vaccine for flu has been approved, and microRNA 2024. So within the short time, 20 to 24, if you see, it clearly says how important and effective is this thing. So in about 4 years, 3 Nobel Prizes have been awarded. That was in 2001 you saw. Now you look at in 2025, actually I can update in 2026, 27 drugs have been approved based on this principle, for various indications. I said that it takes 15 years to find a new drug. Now within a span of about 25 years, 27, now it is 28, 2026, including this flu vaccine becomes 28. And so all these drugs have been approved for various indications. I will not go into that. I will give examples, 2 or 3, how these things work. And of course, we had this messenger RNA, vaccine, COVID thing is also included in this thing. So they have been approved. Why it is so, why DNA has become our RNA. Now the future era is called, is based on RNA. Now, just 2 slides and tutorials on DNA structure. All of you know that DNA is a double-stranded molecule, and each strand will have a sugar, phosphate, backbone. Attached to each sugar is a 4, basic important nucleobases, A, G, C, T, attached through a glycosidic bond here. And there is a complementary strand G binds to C through 3 hydrogen bonds, A binds to T through 2 hydrogen bonds. So 2 DNA, if they have a complementary sequence, they can bind each other together to a duplex. The DNA forms a duplex like this, right? And this is a complementary thing, T cannot bind to G or C cannot bind to A, and this complementary strand, So if you know the sequence of one strand, you can automatically design what is the DNA which can bind to the other strand. So given this kind of primary sequence structure, which dictates its third, so it forms what is called as a double helical structure. Even RNA also forms, it forms loop, it's like you see it is going around, coming back. It forms loop, double strand structures. It's a much more wide varieties of secondary structures. Not only DNA can form double helix, it can also form a triple helix. I will not go into too much into detail. If you have a double helix, a third strand of DNA can come and recognize this. It can bind in the groove, and that you call as a triple helix. Again, this is directed by hydrogen bonding between complementary, base strands. So duplex, triplex, and notorious DNA, it can also kind of tetraplex. And this is a very, very important biological activity, because when cells divide, there is an enzyme called telomerase that recognizes, this tetraplex, which is, it causes that. And telomerase activity is very high in dividing cells. and cancer divides very fast. So one of the methods for cancer drug discovery is through making these kinds of tetraplex structures. If Gs can form tetraplex, 4 Gs have complementary with Cs, and the Cs can form another kind of a duplex like this. This is called as the I motif. It forms tetraplex in a different geometric orientation. So all these things we can use this wide varieties of DNA structures to find out, to make states. Where do you make it, design thing? And that is where this technology has a lot of things. You can make in the initial, untranslated, region. Here you can make a short DNA, about 18 to 20 base pairs. So you can put it, and then the initiation will not have untranslated region, or you can put it in the splicing region, or you can actually put it anywhere here to stop. So for one thing, you can design 4, 5, 6 sequences, multiple drugs for one thing, which is not possible in the case of proteins. You can only one molecule can go and bind. So this has a lot of it, but practically all of them will not be equally effective. You have to do a lot of screening also here to find out for each one of them which is the best region, but all of them can stop this 60 years, 40 years, the ribosome one going. The other thing which happens is that the moment it comes and binds to an RNA, if it is a DNA molecule, you get an RNA-DNA hybrid, then not only sterically it can block this, there's an enzyme called ribonuclease H which can come and cut it. So the idea is to either inhibit this messenger RNA or totally destroy it so that it doesn't make any protein. So these things are possible. So a simple cartoon diagram, this is the ribosome and this is the mRNA, and you can put an antisense oligonucleotide anywhere, and this is the initiation codon. So the ribosome, once it comes here, it cannot pass this at all, or it can get hydrolyzed, destroy the mRNA. So either you make it inactive, numb, or you kill it. That is the strategy for antisense oligonucleotide. You have to do some bit of screening to find out which is the best one. So the principle is so general compared to the conventional medicinal chemistry, I think. Splicing aberrations, I will give you 1 or 2 examples, and I'll just quickly see how I can, proceed to that. So diseases amenable to DNA therapeutics are a number of them. So only different DNA RNA sequences will give you different drug molecules. It is just to make the things very simple. But the moment you say DNA is a drug, that's it. Whatever small molecules, small molecules before they're approved as drugs, there are several properties are important. They must go and bind to your target, they must bind with high affinity, and they must be water soluble, they must have a chemical stability and safety, good toxicity profiles, and they should be easy to manufacture, and they're easy to formulate, simple to deliver. This is the biggest problem. You can make anything, the drug, but taking it across cell membrane into a cell is not very easy. Cell membranes, they protect the cell from undesirable effects, etc. And they must have right pharmacokinetic profile. So even if you say DNA is a drug, we cannot compromise on any of these things. So we have to make DNA conform to all these things. Which can be done is to modify the structure of DNA, chemical structure of DNA, so that it becomes stable to nucleases. Nucleases are enzymes which cut at the phosphate bridge. If you have a phosphate here, ester, they hydrolyze them. So if you want to make them stable to nucleases, one of the easy thing is you change this breast cancer, etc. Although this result is in mice. And then when they made these gapmers, gapmers means change the structures, remove the less sulfur, etc. It was even better. The tumors, and at the same time, the tumor size became even then less. So you can directly inject this into solid tumors. So that was came. Now in 2003, 1997, it took about 6 years to come out with a second drug, approved drug. Now here you see that when you look at this molecule, you can never think it could be a drug molecule. Now here what they have done is, here they have got a OR, chemically modified ones, and then the deoxy thing is there. So they started varying the structure. In the 2 wings you have 2' substitution, and then also they introduced some sulfur substitution, etc. And this was very effective for inhibiting apolipoprotein. This is lipid lowering medications, in high cholesterol diseases, etc. So, and this was the second drug which was approved by, it was called Canamro. It's the same company called ISIS Pharma which developed. In fact, ISIS today, one of the leading person is Prakash, who did PhD in NCL, kind of the third time student. So then came drug number 3, 2005. Now the acceleration started, more and more drugs coming. And here again, it's another gap, phosphates and then 2' modified sugar, everything. This was for McGugan, it is a aptamer. This is called as aptamer. Principle is different. We'll not worry about that. But it is based on the nucleic acid. A third drug was up. Then people started looking more into the mechanism of action, okay? How well they can act. Will they really, the way in which we think it goes and binds, or does it something else, etc. When you look into mechanism of action, I already told you, it can go and bind to the target DNA, and this can be hydrolyzed by an enzyme, ribonuclease, and this, what they found was 4 drugs, formycin, they acted by, these kinds of mechanism, they act. Then the second one is by steric hindrance, simply blocking it, because you put a modification which cannot be hydrolyzed by this RNase enzyme, it stays there and it inhibits the translation. Still, no drugs have been approved based on this principle. And then there is something called splicing. I will come down to that a bit later on. And exon skipping, now there are quite a few drugs are available for this exon skipping. And the latest one for which, siRNA principle for which Nobel Prize was given. And here a double-stranded short RNA is used as a drug molecule. I'll come down to that. So it is not that this one principle works, you have to have several principles. Now this is one of the important areas. So we know that, it's complicated. I will take it to a very simple explanation. We know that we have a gene here, right, for a particular protein. And this gene, whole thing will not code to the protein. Even if 600 amino acids for 200 protein, the real gene might be much longer. Not only that, it has got these interrupted ones. This gene is such a way that this black one and the yellow one, black, yellow region, different regions are there. These yellow ones are called as intron or intervening sequences, and these exons are these ones. So what, initially the gene gets transcribed and it makes a messenger RNA like this. So this messenger RNA is exactly complementary to that. And after that, there is an enzyme, machinery called spliceosome. What they do is that these are the only coding regions, the blue ones. The yellow ones are non-coding. So, if you make protein out of that, it doesn't do that. So then what happens in the cells is that these yellow regions are cut off, machinery comes, yellow regions are cut off, and this 1, 2, 3, 4, 5, they're joined in order to get the right messenger RNA. So from pre-messenger RNA to this actual messenger RNA is a long process. This is involved in all eukaryotic, mechanism. So here the splicing happens, and then this produces the translation protein, which is the right protein to be done. So this whole splicing mechanism is complex. It involves a lot of enzymes, etc. Now, what will happen is that sometimes the splicing doesn't happen, properly. Some mutations will be there. So as a result, when it happens, instead of 1, 2, 3, 4, 5 coming together to make the right protein, one of them gets lost out. Here the 4 is completely missing. So that becomes a mutated protein, and several diseases are there due to this what you call aberrations in splicing. That does not happen, so that gives you a mutated protein here. And this causes the disease. So here, very, very difficult to produce a small molecule drug, because what will it recognize? This is a dynamic process. So only thing is that nucleic acid based drugs will be very useful. Where are these kinds of useful? Now, I'm sure you will recognize this, Stephen Hawking, who died of what is called as amyotrophic lateral sclerosis. So there are several motor neuron diseases or genetic diseases that affect the part of the nervous system which controls, we all move here, muscles and all that. It is happening. We don't realize that. It's all controlled by the, the neuron for these things. Now, any one of those things going back, all your muscular movement, distortion of body, everything happens. So 3 of such things, spinal muscular atrophy, amyotrophic lateral sclerosis, and Duchenne muscular dystrophy. You must have heard of the last one at least, because in India we have several patients. It attacks children in a very, very early age, DMD, and Stephen Hawking was suffering from ALS. At that time there was no DNA therapeutics available, so he could not be cured. Otherwise maybe you could have. Now this SMA, spinal muscular dystrophy, it involves the loss of nerve cells called motor neuron, and is a motor neuron disease. Similarly, Duchenne muscular dystrophy. So what happens is that particularly this SMA, just to give an example, it is based on two genes, what is called the SMN1 and SMN2. In these patients, the SMN1 is defective, and so death of motor neurons happens because this is non-functional. But it has to act with SMN2. Now, SMN2 also is there, but its efficiency is very, very low. It doesn't produce enough proteins, which is required. So, but they found that if you put an antisense drug, and this is caused by splicing mechanism. If you put this drug, an antisense oligonucleotide, suddenly they found that SMN protein is produced in enough amounts and you can cure this disease. This was a really very magic. It's a very long one. I will not show. All I want to say is that, finally you have to get a protein which has 1, 2, 8, 3, 4, 5, 6, 7, 8. This is the right protein. But then what happens if there is a mutation here? One cytidine becoming a uracil. That's all the mutation. Mutation happens in the 7th part of the gene. Because of that, 80-90%, they don't produce this 7. It is not here. 7 is missing here. But so this only 10 to 20% protein happens. But the moment you make an antisense oligonucleotide to this region, CECA, a drug called Spinraza, if you make that antisense oligonucleotide, then this thing comes back, and now you have about 80 to 90% of them, they produce the full protein. You see this 7 is there here, 80 to 90%. So here, only antisense drug will work. Now there are several medicines which have come out, and although number of patients may be less, it attacks children, very, very early stages, and over 3 to 4 years, here this child which has been treated with that, or even in adults, many of them have recovered by, this thing, oligonucleotide-based drug molecules. Now, another one which is for the Duchenne muscular dystrophy, I told you we have several patients, with DMD things. So here, the morpholino-based oligonucleotide work very, very well. Here also is the same kind. You have exon 6, exon 7, exon 8. They have to come together to form the full protein. But because of a mutation here, sometimes this 7 goes away and 6 and 8 comes together. Mutated protein, that is the one which causes this Duchenne muscular dystrophy. And by putting this oligonucleotide 1, everything comes back, 6, 7, and 8. And I hope I'm explaining in a very simple way. It is very clear so how oligonucleotides are important. Now there is another disease called hereditary amyloidosis. Here you can see that this happens because of a protein, because of mutation, it gets misfolded. It starts accumulating. And within about 3 to 4 years, the rapid disease progression starts like this, and then finally, I think motor weakness happens and sensory loss. The median survival is only 4.7 years following diagnosis. Again, there is no small molecule drug for that. But then they found that oligonucleotide-based one, ATTR, it is the FDA-approved siRNA. I have not talked about that. That is a drug. Onpattro is the one. Now they have seen several improvements. So it's very, very, you see the medicinal chemistry, what we know earlier, and now how we can convert, nucleic acid species, the genetic material itself into a drug to address several of these kinds of diseases for which, the things are not there. So this, administered intravenously, targeting, a particular ATTR gene. So you control the mutation by, I think. Now I will skip this in view of time. It tells about how siRNA therapeutics works. I don't know whether I could, maybe just 2 minutes it takes, a small video. And can you, is the audio on?
Yes, sir.
Yeah.
Just a second.
Yeah. Because it's amazing, to see how molecules actually act inside the cells, the mechanism. And so if I take about another 10, 15 minutes, okay? Yeah, I hope it's not boring. Yeah. Or I can just put the microphone here and it will, right? That will do that, yeah. You have a microphone?
RNA interference, or RNAi, is a natural process of gene silencing that regulates gene expression, and is the process that Alnylam's RNAi therapeutics utilize to treat disease. Gene transcription begins in the cell nucleus, where messenger RNA, or mRNA, is produced from a double-stranded DNA template. The mRNA then travels into the cytoplasm, where ribosomes translate mRNA into proteins. Proteins mediate all cellular processes in both the normal and disease setting, with certain proteins involved in the cause or pathway of disease. The RNAi mechanism enables the transient and selective silencing of any gene by targeting mRNA for destruction, therefore preventing the expression of proteins and their function in disease. Alnylam's RNAi therapeutics harness this mechanism using small interfering RNA, or siRNA, which are chemically synthesized molecules that can be administered to silence specific genes. An siRNA is a short oligonucleotide duplex with a genetic sequence complementary to the target mRNA. An siRNA forms a protein RNA complex called the RNA-induced silencing complex, RISC, causing the siRNA to unwind and the complementary functional guide strand to be loaded into RISC. The genetic sequence of the guide strand directs the RISC to the targeted mRNA. The RISC, functioning like a pair of molecular scissors, cleaves the mRNA, causing it to degrade and stopping the synthesis of the specific protein encoded by that mRNA, thus reducing unwanted gene expression. The actions of the siRNA are catalytic, meaning that the siRNA-loaded RISC can target more copies of the same mRNA and destroy them. RNA interference is an innovative and clinically validated approach to specifically target and silence genes that drive disease. Our pioneering science has yielded the critical breakthroughs that have made RNAi-based medicines a reality. Alnylam has led the translation of RNAi into an innovative new class of medicines. Our approved medicines and robust pipeline are focused on multiple therapeutic areas, including transthyretin amyloidosis, ATTR, rare diseases, cardiovascular diseases, metabolic diseases, and neurologic diseases. We're pioneering what's next and working to bring RNAi therapeutics to a broad range of rare and prevalent diseases.
Okay, so this is a company, Alnylam, who introduced this, and Vasant Jadhav, who was my PhD student in NCL. Today he's the Chief Technology Officer of this company, and I think Manisha, she worked around the same time. Now, whatever you saw that, this is a thing. See, this siRNA is double-stranded RNA. It comes and binds to an enzyme, and this enzyme separates the two strands. So it is what is called the antisense. The sense strand is removed. Complementary, the red one is the antisense strand. Once it comes here, then this one is loaded onto another machine, and miRNA comes and binds to that. So these things are designed in such a way that they're targeted towards a particular messenger RNA in which they defect. Once it happens, then it cuts this messenger RNA, this enzyme cuts it, goes away, and this strand is still bound, that gets recycled here. It comes back, comes back. So many messenger RNA things it slowly destroys. That is called as the RNA therapeutics, which is now becoming very, very important. Several drugs are coming out based on that. Okay. Now the biggest problem here is how you deliver this short siRNA. RNA, single strand RNA is unstable. It gets cleaved. But if you have a double strand, short ones, 20, 23 months, there's a natural mechanism, but they're very stable. So this Alnylam company, they designed what is called as a lipid nanoparticle. So they used, wide varieties of lipids, which are, having different kind of ammonium groups, because these are called as, in endosomes, these liposomes, they go, and then they have to liberate the RNA from the endosomes outside, and that is done through a pH process, very complicated for those who do not understand that. But that pH is controlled by using at least about 6, 7, 8 different kinds of lipids. It's a concoction. Why it is important is that once they developed this lipid nanoparticle for delivery of siRNA, same lipid nanoparticle was used for mRNA vaccines, which gave in the COVID time. See, they didn't have to invent anything. This is available. So they packed mRNA into the same things and they use it for that. In fact, the acceleration, to the application became very, very fast. That's why this mRNA, I think came very good. So this is with respect to how to deliver it, lipid nanoparticle. And another strategy to deliver, I will tell you about two strategies, is, if you have these hepatocyte cells, in the liver cells, there are a lot of virus attacks, liver diseases, they come. Delivery of drugs into liver cells is very, very difficult. And so they are not, particularly for hepatitis C, there are not very many small molecules. Very recently, one small molecule was drug, but before that, these RNA therapeutics came. Because these liver cells have receptors, you see these receptors? This is called as acyloglycoprotein, ACGPR, It has got 3 of those receptor hands, so they can bind. If you have this so-called N-acetylgalactosamine, N-acetylgalactosamine is a carbohydrate derivative, 3 of them are there, they can go and bind to the receptor, and once they bind to the receptor, that is taken inside. So siRNA is taken inside and it happens. So now this is the one way in which you can deliver siRNA. One is through lipid nanoparticle, other is through this protein thing. Now siRNA, I will not go into the mechanism. Now it can, as I said, RNA therapeutics is a wide area. You can, for example, look at kidney diseases, hepatic diseases, pulmonary diseases, metabolic diseases, neurological diseases, hemophilia. So each one of them, you see, these are the drugs which have been approved based on the siRNA. The principle is so general, that is the importance of this compared to small molecule therapeutics. So all these things are approved drugs, either given through an injection through this, lipid nanoparticle or with the other ones. Now, I will not go into that. There is a simply called a microRNA. Before RNA comes into the cells, there is microRNAs. They are in the non-coded region, microRNAs. So they come out and they form certain structures which they get cleaved before they come. So now people have designed to target the non-coded region also. And this is still, nothing has come based on this disease, but this is a new mechanism which has come, and for which Nobel Prize was given for this microRNA. Now, this is RNA therapeutics now. There are 14 molecules of antisense oligonucleotides, 2 aptamers, 7 siRNA-based drugs. There are already 4 mRNA-based vaccines are there. But some of them they find, and they have been withdrawn also. 3 drugs have been withdrawn. So 1998, 1 drug, one, and then slowly now I think it is increasing. 2023, just after COVID, that 5 drugs were approved. So there are a total of about today 27 or 28 drugs approved, and this is, it's a folded, RNA thing. These are the various companies which have, FDA approved one. So it goes, this is 1998, 2004, 2013. It is based on, which drug was approved in which thing, etc. So it's a very successful, concept which has come out. And these are the company, for the indications for which these drugs have been approved. Very complicated slide. But just to tell you that this is now a successful venture. Something, concept came only in 1887 or 1890 something. First drug came in 98, and now in the last 25 years, there are many of them for which there are no small molecules are available. So now there are several drugs which are currently in clinical trials, for varieties of diseases, cardio, metabolic, cancer, CNS, respiratory, because the principle is, so general. So you have to find the corresponding targets. This I already have shown you, a very successful thing. Now I'll just take another, 5 minutes to tell about where do we stand. Why am I talking about this, nucleic acid-based drugs? Now, much of the drugs, common clinically approved drugs, whatever I've shown, they're all only because of chemistry, chemically modified ones, nucleic acids. These RNA, GNA, these 2' methoxy, 2' fluoro, 2' O-methoxy-ethyl 1. And then you have some modified bases are there, modified backbones, phosphate became phosphorothioate, then became morpholin oligonucleotides. So much of them are based on one of these kinds of, modifications. And this I already talked about. This is the N-acetylgalactosamine. This is the one which binds to the hepatic, that protein and gets transferred inside. Now, this is again, to give you that how general it is. Now people are concentrating more on the neuronal diseases. CNS diseases is being very, because there are not, again, very many drugs available for the CNS. And here they found that if you simply attach a C16 chain, that goes to the neuronal cells, crosses the blood-brain barrier, and, so some work is being done. So these are the different kinds of RNAs which can be, microRNA, non-coding region, long non-coding regions, ribozymes, so many things are being targeted. So there's plenty of human genome sequence has thrown open so much. Now, in the next 5 minutes or so, I will tell about where do we stand. I mean, we have not gone to that stage. So we started working on a kind of molecule which are called as peptide nucleic acids. You remember I told you a very simple one, ethylene, diamine, glycine, to which nucleobase is attached. In India, if you want to work, your choice of problem is very important. You must take a very simple problem, at the same should be interesting, because we can't compete with all those big giants. So we went to a molecule which is not chiral, which doesn't have anion, simple peptide, we can make it. So the phosphate, now these are called as the peptide nucleic acids. So you have a simple backbone, unsubstituted, no chirality, no organic chemist will get interested in this molecule. So we don't have much competition. And simple amide chemistry, any peptide machines can make it. Now the good thing about this molecule is that if you have a PNA and now attach these bases, they can bind to a complementary DNA exactly in the same way as one DNA would bind to another DNA, or one RNA would bind to DNA. So it binds exactly in the same way. This is the PNA and this is the so-called DNA. It forms a duplex structure. Not only that, it can bind in different fashion. Don't worry about this parallel, anti-parallel. The important thing is that this PNA-DNA complex is much stronger than the DNA-DNA complex and the DNA-RNA complex. So if there is a DNA-DNA complex, you put a PNA, it can go displace the DNA strand and bind it. So it can find more targets, is possible with the PNA molecules. They have a high thermostability. Second, another important thing is it is neither a peptide, because peptides means 2 alpha amino acids are involved. But here you have ethylene, diamine and a glycine. So it is not a peptide, which means it is stable to all proteases. And it is not a nucleic acid, it is stable to all nucleases. So it has this advantage, not only stable, in vitro stability, a strong hybridization. See, if you take DNA, it requires a lot of salt because both the strands have negative charges. But here, there is no negative charge, so it can hybridize at low salt concentration, the cellular level concentrations, and it also shows sequence-specific binding. If there is one mismatch, the stability goes down like anything. So it is very, very sequence-specific. So all these advantages are there. And also, it has, the toxicity is also very, very low. That was. So it was like a dream molecule for a medicinal chemist to start with. But it has other problems. It didn't come up very much. But we have been doing a lot of chemical modifications. The objective is chemical modification of PNA to overcome. It binds to DNA and RNA. Can you make specifically something which can bind to DNA or RNA? And also it has this directionality problem. So we thought that we would look into that in a chemical way. And I think, maybe one more I have to put. Yeah. So this is where we started, 1995 to 2005. That is my NCL days. And you recognize Manisha, who is in the audience. And so it started with, Gangamani, who made, So what we thought was, let us make modifications. Let us make a PNA, which is a cyclic molecule, look like DNA. Let us introduce the chains. So that you can do by cross-linking, 2 of the carbons, either in the backbone or from backbone to the side chain, etc. So if you take the simple molecules, you can make all these kinds of varieties. It was a 10-year research, sort of I have given in one slide. So each one modification was one student, and also this molecule becomes chiral, 2 chiral centers you're introducing, and you had to, of course, worry about the chirality resolution and all that, but we did a lot of stereospecific, We had proline to start with, and Ganga, she is now in a big job in Abbott. Manisha is here. He is a faculty NICER. And Meena is a very, very big name in Boston. She's working on a lot of locked nucleic acids. And Sunil is somewhere in Hyderabad company. Pallavi is again in Boston. If I go to Boston, a lot of my students are there, and different companies. And then we could even make, what you call a 6-membered ring. And Praveen was with AstraZeneca. Now he has moved to Biocon. And Govind is a faculty in things. So we made all these molecules, make it chiral separation, look at the DNA stability, TM, etc., synthesis. Sometime we had to do a resolution. But still, after so many years, I could not give a general rule, whether this is better, this is better, this is better. I could not predict it. We are getting a lot of mixed results. Till Govind decided, instead of joining the two things, I will only concentrate on one bond. There is one CC bond in PNA. Let me make a cyclohexane ring or a cyclopentane ring. Only worry about one bond. When we made that, we found that these kinds of chiral, the cyclohexyl RNAs, you can have a cis trans, etc. And he found that this stabilizes, binds mostly to RNA but not to DNA. So after about 10, 12 years of research, we found one general route what we could do with PNA. But we produced a lot of students. They got experience in RNA. And many of them have been working today in the DNA area. Then we went back to the acyclic backbone because synthesis were very difficult. And again, Rupa, Deepak, Deepak was in ICER, but Rupa and Satish was in ICER. Rupa worked in NCL itself. So I think so, yeah, I've forgotten. Was she in NCL or ICER? Yeah. So here we thought that we'll just simply add a cationic one because if DNA is anionic, let us just add a cationic one so it binds better. So we went to putting a aminomethylene group here at the gamma position or at the alpha position, and or you can make even a stronger one, guanidinium, simple chemistry modifications. And then we know that many drugs have 20% of the drugs have fluorine in them. Fluorine gives very metabolically very stable compared to CH bond. CF is stable, and it also goes inside the cells better. So Satish made a lot of fluorinated PNA molecules, putting fluorine, like for example, we have only 2 fluorines per monomer. Here we have 2 fluorines. Here we have, 4 fluorines, so we can control number of fluorines. And so what we found was that these things increased stability. PNA is very poorly soluble in water. These things went better. They hybridized better, and cell permeability was better. And these things also we found that the fluorinated one went inside the cells fantastically better than the normal one. And then we started, making conformationally constrained but without introducing a chiral center. Jump dimethyl groups here, and they show some interesting properties, but they're much more hydrophobic. And so these are the kind of modifications we thought about that. And this just shows you how do you investigate that. When you make it, you label with a fluorophore at the end, and then look at how they go inside the cells. This is called as DAPI, which goes and shows the nucleus, the stains in the nucleus, the blue dots. And these are the carboxyfluorescein PNA molecules, they're going inside, in the cell, all these fluorescent ones. And if you superimpose these two, you find that you can see the blue and you can see the green. What it means is that it is going not into the nucleus, it is in the cytoplasm. If both of them are co-localized, you will see an yellow color, So we could see, and we can quantitate the fluorophore thing, see how much if it is going, etc. So we were very happy that we could make PNA go inside the cells better. These are similar results with cationic amino acids. But here we found that in the HeLa cells, it is going right into the nucleus. It is not going to the cytoplasm. HeLa cells, it is localized in the nucleus. And as chemists, we can also could see whether R is better, S is better. In the alpha position, R was better than S. Here, S was better than R. So we could do those things. And guanidinose ones actually went like mad. They were uptaken in the cells in a huge amount compared to the amino. The guanidinose were going. So we could do this kind of structure-activity relationships with that. And then, inspiring from the GalNAc one, we took, siRNA, I showed you that GalNAc can take it inside the cells. So we made a peptide nucleic acid molecule to which we attached this GalNAc. This is in collaboration with Mano. He's the one at Al-Nailam who is really the king of siRNA. He's a very highly respected medicinal chemist. He has put 6 siRNA drugs into market. So he said that let us do with the PNA. So we did with PNA, we attached in the end, but then the PNA also provides, instead of putting this complex one, we could simply put a lysine chain attached GalNAc. And then we found that these kinds of molecules went inside the cells much better than the triantennary, which is much more difficult. So we could, we could see that these ones were, taken up much better inside the cells. So you can do this kind of chemical modification. After having come to that, now we have to find a target and work on, that PNA. So that is the one. So now I'll quickly, in the 2-3 slides I think I have, Then we went into a different dimensions. You have a PNA, it has all these bases, it can bind to one DNA on the top, that's it. But if you look at PNA structure, an organic chemist, I could think that I can attach one more base. In the same nucleobase, I have one base here and another base. This we called as the Janus PNA. It has two phases. So this PNA backbone carries one sequence on this side, one sequence on that side. And you can attach it through a triazole, or you can attach through an amide linkage. What this means is that if you take that kind of a PNA, which has one backbone, two phases, if you take that, then you can silence one gene on one side, and if you change the sequence, you can do that. So with one PNA, you can hit two genes simultaneously by two different sequences. So we thought this would be a very interesting applications. So this is something which you cannot do with DNA at all. Only with the PNA, chemistry allows us to do that. So these are the Janus PNAs, and it has got two phases, and so we went to that. I will not go too much into details, just to tell you what is the importance of that. See, you have, you take one Janus PNA, so here on poly C, poly T you have, so you can prepare one DNA on one side, this is the tertiary amide one, this is the normal amide one. So you can make two individual duplexes, one on this side, one on that side, but when you have both the DNAs, it can form two duplexes. And how do we know is that you can do, for each duplex, you can do a TM. You measure absorbance at the, as a function of temperature, and when the DNA separates out, it gives a peak in the UV melting. So now here, both of them, they gave only one peak, and here you can get two peaks. Not only that, this stability of this was much higher than these two, so we could do that. And in this case, we did with a chiral stereochemistry. Here there's a chiral center. We can do with R stereochemistry, and we can also do with the S. So here we found that the R double duplex, was much less stable compared to the S1, 73, 69, 57.38.7. So we now have these new kinds of ones. I just gave you two examples. Now we have a plethora of these Janus PNAs with a wide varieties of linkages. So we've been able to make, this called as a double duplex. And you can make a triplex. So one triplex, then you can add two more strands. It becomes what you call the triplex of a duplex. 5 strands arranged together. And then you can have a tetraplex. We know that G forms a tetraplex. I told you, if you take this and put a Janus PNA, then you can add another one. So you have got 8 strands. It is called as a tetraplex of a tetraduplex. It is possible. If you put silver, cytidines can bind, and then you get duplex. So we have been able to generate number of what's called polyplexes, which also have important this thing in what you call the material chemistry. Now these ones, we are not very sure, the Janus ones or the modified ones, whether they go inside. So this is a Janus PNA, have G's on one side, C's on one side. I take a control, only G or only the other bases. They are not going inside the mix AEG fine. But compared to these things, the Janus PNA is going inside. This is a very, in both MCF and NH cells, it is going very well. Now, there are no drugs still approved on the PNA. Still it is in the development process because PNA is not very easily accessible to biologists. Very few companies make them. In our stand, we all make everything from the start. Now only some few companies have come out, and very recently, a lot of PNA have been used for targets to address several of them, in vivo, in vitro, for different disorders, observed effects. I think in the next 5 years, a lot more things may come from PNA, and if that happens, we'll be very happy because much of our work may come into the limelight. So that is about it, my formal thing. I'll just show you 3 or 4 slides. Do you people recognize what is this? ISER Pune, just next door. I think the Ahmednagar College students may not have known that. I know this is the one which, was built in the NCL, thanks to NCL who gave land. And this is our main building. And I used to have 2 things, how this land was looking, Google map, before ISER came and one after ISER came. This is the latest one, we have, hostels, these are the faculty quarters, hostels. I think some of them are being built here, it was taken at that time. So this is ISER Pune, it took about 10, 11 years to build. And this is the ISER Tirupati, which was all built in about 4 or 5 years because of the experience of a contractor, what I got here by building things. we could do that. And this is architecturally, it's already got lots of awards, particularly this building. This architecture is very, very good. This is the auditorium, this is the administration block, and here you have all the hostels. Everything came up within about 4 years. We could do that, including 2 years during COVID. All the hostels were built, and there's beautiful ponds. This is a very big land compared to Pune. This is about 250 acres. So there are a lot of land available for future expansion of, ISER, second phase, third phase, etc. So this is the ISER Tirupati. And I was telling you, this was the architectural wonder where about 50 different equations, names have been built on a concrete thing, inscribed. It was architecture, it was very, very difficult. So this is the photo op, anybody goes there and you can take a student and ask him to identify which equation is it, etc. And I must just tell you 10 seconds a story. I had to give in writing what are the things which has to be inscribed. I took everything from Aryabhata to everything, whatever came to my mind, equations, everything I gave. I sent to the CPWD engineer, and within half an hour he said, "Sir, Schrodinger's equation is not here." Can you imagine a CPWD engineer talking about Schrodinger's equation? Then I said, I looked into that, it's a long one. No, sir, this is a short one. Hep psi is equal to, the short one, and that is somewhere here, we inscribed that. It would be nice to have such, team. So I had a fantastic CPWD team working. And this is, you can see this. Does it look like a periodic table here? This is the auditorium wall. Somebody said this is the biggest periodic table on the earth. So we have made this, and the colors have been matched to the color of the salt. In the night it looks very, very nice. This is auditorium. So we have a periodic table. We have all the physics captured as DNA. Now I only want them to put a DNA thing. Then all science, physics, maths, biology, everything has been captured on the campus. That's a visual one. So much of the, things what I did in ISER Pune, the Janus PNA, etc. team was, students, and actually initially some of them worked in NCL before our lab became complete. And these are the students who joined with me in ISER Tirupati, and some, are not sure about the publication thing. So I had to thank, the various organizations who helped me in my research, and that's where, and thank you. I am known for taking more time, sorry about that. So you still have some question and session, I'll be happy to. Okay, fine, yeah. Okay.
Thank you, sir. Thank you very much. I'm very small to talk about the research, but one thing which, as a student, we have noticed, sir was excellent guide since our college days. We were remembering his name through our seniors and many other people, and at the same time, he was excellent scientist, developed into excellent administrator. You have seen that photos also. And also the excellent speaker we have seen so far, in spite of having Padma Shri and so many awards, the way sir has made that topic a simple way that everybody can understand what is RNA and DNA, what we can connect with our 12th standard books also. So we are really thankful to you, sir. And I would like to request everyone, we can give stand-up ovation to the sir, because he's Padma Shri awardee at the same time, very big personality. Thank you, sir. So with this, we will move toward the felicitation program. So I request sir to be on stage, and we have few things to felicitate, like Puneri Pagadi for you, and then I request Manor sir as well as Bhushan sir to please be on stage. On behalf of NCL, I request Ramanna sir, Mahesh Kulkarni sir, and from family members also I request
Yeah, sure.
Thank you, sir, and I thankful to all the guests. And with this, we would like to move toward the end of the program. But before we go for vote of thanks, we have Saurabh with us. So as we discussed that, somebody from family can also share their experience. So I invite Saurabh. Yeah.
Thank you all. I'd like to thank the organizing committee for this, and especially Dr. Manohar, Dr. Bhushan, Dr. Ganesh. Thank you for organizing this. It's really special to us. Uh, for those who don't know Dr. Khanna, he was a great teacher. He was a great mentor. He he would help people a lot, and you could reach out to him for any of your issues, and he would be more than happy to help you out. And I'd like to share a few story probably with you all. So before all the achievements that he had, he was a young man who listened to his father, and his father wanted him to run a dhaba. Back in late 70s, in 70s, education and getting a job was not something that was preferred. People used to prefer doing jobs, preferred doing business, right? And that's what his father wanted him to do. And he did that. He did that for 2 years. For 2 straight years, he was running a dhaba. He served food to people, and in those 2 years, somewhere, he realized that it was not his calling. He wanted to pursue something else, and he rebelled. He came back to Nagar. He proceeded with doing his graduation, master's, doctorate, and then eventually went on to work in the UK and then in US with a Nobel laureate. And with all this, right, I think he had great passion for teaching. He came back. He started a course. He was pioneering that course, and I vaguely remember him telling me stories about how he would meet the education ministers, authorities at that time. I mean, imagine somebody in his 30s trying to convince people, to make them understand how his course is different than pharmacy. And I think along those lines, with the struggle that he had gone through, I think he achieved that. He went on to, and his course did quite well, right? I mean, the results speak for themselves. His students, the companies would prefer his students, when it comes to recruitment. So I guess you guys, you students, his students specifically, you are his legacy, and you continue to make him proud. At home, he was a jolly man. He had a lot of stories. You could talk to him about any topic, and he would have answers for you. And it didn't matter. You could be his student, and you can share your personal problems with him, and he would ensure that he could help you out. And I'd like to conclude this by quoting one of his favorite quotes, and I remember this. He would say this a lot. I'm sure if you were in his class, you must have heard this. He would say, "It's not failure, but low aim, that's a crime." And may we all aim high. May we all continue to make him proud. And thank you all.
Thank you, Saurabh. We remember one message, like when Sir was having a treatment, and we everybody wanted to meet him. He was telling, "Don't worry, I have Saurabh with me, and whenever I will feel that I'm okay, come back to Nagar. We will definitely meet." So we know that you have done a lot of things for sir as well as all the family members. So before like we proceed for vote of thanks, I'd like to request, can we have one like photograph with our chief guest and with all the audience with the family in front, so that we can cover this, particularly all the students as well as faculty who came from along like from the Ahmednagar within one photo. Yeah. So I request sir, you can please take photo from here, and family members along with the chief guest can be at the center. And I request everyone can stand up for the photo so we can have, and after that, yes. Saurabh, family members can join for the photo. I request those who are on right and left, please little bit come to the middle so that we can cover everyone in the photo. So madam has also come. Thank you, madam. Please come on forward. Bhushan sir. Yes, yes. Thank you all. And with this, we are moving towards end of this program, vote of thanks. We try to make it as much as possible as per the time and schedule. So I would request Yogesh Munotji. Before I announce that, I just like to have a small announcement that we got small sponsorship from Dr. Bapu Gowde and Dr. Yogesh Munot from their company for hosting this event. Yeah.
Respected guest and all, it's a very emotional moment. Hardly we see anybody like Khanna sir after the words, after he left, something like this is happening, and the best talent is here on this diet who at desk today. They have given the lecture for this, and it's really, we feel privileged. Personally, I feel so many stories are there, but because of the time restrictions we cannot share. But I would like to thank all organizing committee. First and foremost, extended heartfelt thanks to our chief guest, Padma Shri awardee Professor K.N. Ganesh, for his inspiring and memorable memorial oration. His words have enriched this occasion and will remain a lasting tribute to the legacy we have gathered here today. We are sincerely grateful to the director of CSIR-NCL for generously hosting this event and for extending institution support and hospitality. Our special appreciation goes to Professor Bhushan sir, as well as to Dr. Manohar sir, for their visionary planning, guidance and dedication in bringing this event. Special grateful acknowledgement and thanks to BPH Nagar College Society for the institutional support. We also express our sincere thanks to Mahesh sir, Ashok Giri sir, Ramanna sir for their meticulous structural coordination, Our appreciation also goes to Dr. Tejas, Shri Bendale, Shri Sanjay sir, and the entire event management team for ensuring and the smooth and seamless arrangement of the venue as well as today's function. We would also like to acknowledge the invaluable efforts of our YouTube live streaming team, Mr. Jagtap, Mr. Avinash, Askar, Javed, Raju, whose work enabled many of our well-wishers and alumni to participate virtually from across the location. Our heartfelt thanks to Dr. Sunita Barve and the NCL archive team for their valuable contribution in preserving and the presenting the institutional and scientific legacy associated with Professor Khanna sir. We also appreciate the dedicated efforts of Sri Chitanvis and the NCL security team for ensuring the smooth and the secure conduct of the program. We are also equally grateful Dr. Gati, Sri Balaji sir, and the NCL canteen staff for their warm hospitality and for taking care of the needs of all our guests. Finally, and the most importantly, we express heartfelt gratitude to all our distinguished guests, colleagues, alumni, students, and well-wishers who have joined us today, both in person and virtually through YouTube. Your presence, participation, and the affection have made this occasion truly meaningful. On behalf of the organizers, I once again thank you, each and every person who has contributed directly, indirectly, to making the gathering of fitting tribute to the life, work, and enduring legacy of Khanna sir. Thank you for being part of this memorable occasion. Thank you.
So with this, here we declare like end of this program, but not the legacy. We will be continuing with this one. There are many programs are planned in coming year. So please be tuned. And we are again really very thankful to Padma Shri Professor K. N. Ganesh sir for accepting our invitation and coming here and enrich our students as well as alumni for this function. Thank you, sir. Thank you very much. Thank you, sir.